In today's lecture, we discuss about this biotechnology derived products. Already discussed that this is not mentioned here as syllabus. This is the content other than mentioned syllabus. This biotechnology is again a very important types of that formulation for a drug. So we will go in detail with this biotechnology derived drug products. So when we talk about these biotechnology derived products, many drugs are now derived by these biotechnological products. Many vaccines are produced this biotechnology uh, by biotechnological way. So there is a large productivity is there. Okay, no animal is involved, so all these are the advantages of this biological processing. So which parameter we will consider, that now we will see. Number is the bioreactor. Bioreactor operation is very important. Then filtration, then centrifugation, chromatography, viral clearance. So this bioreactor operation, that is the stir tanks we use. So the rate, aeration rate, hydrostatic pressure, agitation rate, mixing time, all we have to study during the formulation in pilot plant, also in the scale up technology. So when we go with the scale up, the imperial rate we have to study. We have to study the aeration time. So that's why proper oxygen level can be maintained. Then the hydrostatic pressure, then agitation rate, proper agitation is there for the mixing. Okay, then the mixing time. So initially in the pilot plant, we are using a small bioreactor. So its impeller rate may not be sufficient for your large scale bioreactor. So we have to maintain, we have to study, we have to change the process. So that's why we get a product with that predetermined standard, or we can say that with that predetermined yield. Here we have to give, we receive the product. So we can say that a yield. Then when we talk about the filtration operation, what is the membrane? What is the volume for that filtration assembly? Operating time for how many times? Temperature of that filtration assembly? What is the flux rate? What is the protein concentration? What is the solution viscosity? If solution is very viscous, we have to change the filtration assembly or the filtration unit. We have to add more suction rate. Okay, then that flow rate we have to reduce, so that's why it will not choke. Then permeate flux, all these things we have to study. Then other variables, when we talk about of this biological product in relation to that filtration, are the length of the fibers, means the fibers which we are using to prepare that filtration material. Then its diameter, fiber number, then density, then viscosity of your culture. So from these variables, scale up parameters such as the wall shear rate and its effect on the flux are derived. Chromatography means we have to identify that product. Okay, generally viruses or the bacteria are used to develop a product biotechnologically. So in that chromatography, generally we use gel chromatography. So gel capacity, linear velocity, buffer volume, bed height, temperature, cleanability, gel lifetime, pH of the elution butter buffer, then conductivity of the elution buffer. All these things we have to study. In chromatography, we get different bands. According to that bands, we can determine that which product is there. Then we will isolate it. We will use different solvents, okay, to get a product. And in which solvent or in which by using which procedure we get a product with high yield, we will choose. Then viral clearance, it is somewhat very important process when we talk about the biological products. So we have to neutral all the viral, or we have to remove all the viruses from that product. Okay, viruses means what? The product, the viruses which may be harmful. Many times, if we prepare a vaccine, viruses are used. They are neutralized one. They doesn't contain any viral substances, okay, which may be harmful. So we have to remove it very carefully. That vaccines may be a empty boxes of that virus. Okay, so it is very important to remove that viral material, viral, that is very the danger material from that virus. So it is also we have to scale up. Then when we talk about all these dosage form, what is the development milestone? Number one is the which product we have to market. That is first identified. First we identify the product which we will prepare. Then we develop a process. Okay. While developing a process, some critical steps is determined. Packaging parameter is determined, and then that product is developed in a pilot plant. Okay, after that, that product is go into the scale up. Before going to that scale up, its suitability, its proper report is studied. In that scale up, number one is the how, at which site, what will be the initial requirement, what will be the initial that manpower required, equipment, which equipment we will use. All these are determined. It is then installed in that scale up unit. Okay, then we will develop that scale up report. After that product is developed, we will submit that new drug application. Okay, if it is NDA or ANDA, abbreviated new drug application is there, or somewhere there is the generic is there. So if it is a new, then we will go with this, or generally we will go with that approval of FDA, that this is the formula we develop and this is the result. FDA is approved. Okay, we have CDSCO in India, in US they have the FDA. Then there is the manufacturing validation. All these record will be given to that licensing authority. Okay, and after that product goes into the market. We go with the production startup. All these in nutshell are the process for the development of the products. Okay, these are some questions. These are the references you can refer: Lachman, Encyclopedia, and Scale Up Technology, Marshall Decker book. Okay, so with this